You ready, Chad? Just like class, everybody quiets up. Welcome to the inaugural Human Bioenergetics Lecture. My name is Scott Trappe. I'm Director of the Human Performance Laboratory. And I want to welcome both our in-person audience, and we're doing a live stream for our alumni and friends around the globe, so welcome to all of you as well. And I'd also like to start off by thanking a few people. I want to start with Ball State, the continued support and infrastructure support we have at the lab to continue to do what we do and then host events like this. We're at 58 years and counting, so kudos to Ball State for all that generous support over the years. And for today, it's a big team effort to be able to do this. The infrastructure at Ball State as well. Our media team came out and helped with this. The College of Health, Dean Rutledge is here. The chair department, Lindsay, and our new chair, Paul, being supportive early on of this endeavor. And also then the HPL team that it took to pull this together, to fill out all the paperwork to get the venue. Particularly, I'd like to thank Angie, Sherry, and Gary behind the scenes. They were doing all the legwork to make this event possible here today, so thank you to them. So today we're here to celebrate science, getting back together, interacting, but we're also here to celebrate the special occasion of a birthday. Bill Fink celebrated his 90th birthday on Wednesday. We began our lab celebrations yesterday with cake and a nice dinner last night. You're going to hear more about this, but Bill has been a longtime supporter, a big-time contributor to help make the lab what it was and educate a lot of us students, me included, along the way. Congratulations, Bill, on being 90 and all the contributions you made. We'll talk a little bit more about that as we go along. And I'd like to acknowledge the support for today's lecture. It's primarily driven by a fund we set up prior to the pandemic, the David L. Kostel Fund for Excellence. The idea with this fund was to enrich student experiences like this, and we had some of that this morning where the students had some protected time with our external guests, which is always a good opportunity for everybody. So we're sort of past the silent phase of fundraising for the Kostel Fund, or leadership phase, as I found out on Friday night during our capital campaign. And if you are interested in contributing to help us grow this fund to do more events like this, you can contact myself or Dave or our foundation officer, Ryan Vaughn. So with the idea of enriching student experiences, I've asked our senior doctoral student, Chad Skiles, to come up and introduce our speaker today. A couple comments about Chad. He's a senior student in the program. He comes from the Pacific Northwest. He moved him and his family during COVID and made it here in the night, skidded on 2 wheels. He was a college runner, and he's turned triathlete. He's got 2 wonderful swim coaches at noontime during the day. He's made wonderful progress. I have no idea how that's happened. Chad's been an outstanding student in our program. He has a wonderful wife, Kate, 3 young children. He's a triathlete. He's a step ahead in his progress with the program, in his writing. He's been a huge contributor to the motor pack efforts and leading his dissertation. And for those of you out there on the live stream, Chad is currently in the market for a muscle biology postdoctoral opportunity. He's already has a couple opportunities. He's going to interview here soon, but we always welcome more opportunities for our students. So please welcome Chad Skiles.
Thanks, Scott. Yeah, my pleasure to introduce Dr. John Kirwan. He is from Ireland. He got his bachelor's from the University of Limerick and then came to the United States and got his master's from the University of Massachusetts. He then came here to Ball State University. He got his PhD in the Human Bioenergetics program under Dr. David Kostel. He's had multiple notable positions in his career, including he was a professor at the Case Western Reserve University School of Medicine. He was also a director of the Metabolic Translation Research Center and professor of the Cleveland Clinic. Currently, he's executive director of the Pennington Biomedical Research Center and a principal investigator of the Louisiana Clinical and Translational Research Center. He's a big name in our field, lots of achievements. He's a leader in the biomedical research program focused on diabetes, obesity, nutrition, and exercise. He has generated over $60 million in grant funding, primarily from the NIH. He has over 286 scientific publications, and from notable journals such as the New England Journal of Medicine and Diabetes Care. He also has a family, wife named April, 4 children. And with that, please join me in welcoming Dr. John Kirwan.
Thank you, Chad. Thank you, everybody. Let me get my bearings here. So this is the title of my talk, and I just want to take the initial opportunity to say how much of an honor and a pleasure it is to be here today. I was reminded of the class of '87. It's been a long number of years and a lot of water under the bridge, but this experience here at Ball State with Dave and with Bill and with all my colleagues has really laid the foundation for what became a very interesting career, and I'm going to share some of the science of that with you. But it's also an opportunity to say thank you to Dave for the inauguration of this Human Bioenergetics lecture series. I'm sure that there will be many of these to come in the future, and certainly you have lots of alumni who will be willing to dedicate funds to this in the coming years. Bill, thank you in particular for all that you did. Part of the reason for being here today is to celebrate your 90th birthday. It's an occasion in and of itself, and it's a pleasure to share that with you. I'm going to take this group on a journey right now through some science. I'm going to talk about metabolic disease. So this is a lab that's focused on applied physiology and performance, but today we're going to segue into an area that is of more clinical relevance. We're going to talk about things like obesity and diabetes. We look at examples and studies that show how exercise can be used to treat type 2 diabetes. We've got some new treatment programs that I would like to touch on and make everybody aware of. Great excitement in the diabetes and obesity field. And a public service awareness campaign that we originated at Pennington Biomedical a couple of years ago that has had a profound effect not only on public awareness nationally, but at the state level and some of the programs that were developed there. This is my disclosure information. There's nothing here that will be a conflict today. This is the Pennington Biomedical campus. It's a 220-acre campus in tranquil Baton Rouge, Louisiana. We have about 750,000 square feet of research space, an amazing facility, a great story. The foundation of Pennington was based on a gift from an oilman, Doc Pennington, who struck oil north of Baton Rouge. In 1983, he was experiencing some illnesses and he had some health problems, and he was self-medicating with nutrition supplements primarily. And the state approached him to make a gift, as we had a call today for Dave's fund. But this was a call from the state to a very wealthy individual in Baton Rouge. And Doc wrote a check, single check for $125 million in 1983. About $350, $400 million today. It was cash, basically. He kept all his money in his checking account, so that was a good check to get. And his only request was to build the biggest and best nutrition research center in the country. And that has been done. And the program grew from there in the mid-'80s, founded by George Bray, who was a leader in the obesity field, followed by Claude Bouchard. And today, I have the honor of following in their footsteps to lead Pennington Biomedical. I'm going to talk science now, and before I forget all of the people who actually do the work here, I want to acknowledge the people in the lab and the people who I have collaborated with, both internally at Pennington and externally around the country. Chris, so when I moved from Cleveland in 2018. We had a big research portfolio. I brought 15 people from Cleveland to Louisiana, and today I still have 4 of them still working with me. The rest graduated, moved on, and here we have Chris Axelrod, who directs the lab, Wagner Dantes, who's a postdoc, and so we're looking at postdocs here. Chad, make note here. Wagner just got his K-99, which is the big NIH training grant that every postdoc aspires to. So we're very proud of that accomplishment. Elizabeth is completed her PhD at Case Western and came to Pennington, is now a postdoc on our training grant. Again, I want to emphasize to the PhDs here, there are training grant opportunities at Pennington. So we've got Kath, who runs the lab, and then Liz. Liz Hines is a great story in her own right. A master's student from University of Louisiana, came to the lab to work on a work study, decided to do her PhD, and just last week got her F31, NIH F31 grant funded as a PhD student. So a reminder to the PhD students, there's opportunity for you outside of the funding that is provided by your mentors. You can apply for NIH grants, and very competitive, but Liz just got hers last week. Balora's a senior scientist, and I want to highlight Jake. Jake May is a former student of Jake House, who is a former postdoc of the Kirwan lab, and is having a very successful career at the University of Michigan. And you all know that Jake is a graduate of the HPL. So there's a bit of lineage here. And I'm also happy to say that Jake won, that Jake too got his K award just a month ago and is now on the NIH track. Candida is a great example of another postdoc with me who got her K99 a year ago. She's up to her R00 on that. So this is a very high-performing group and have been very successful. And partly because of the opportunities that get presented in places like this. I talked to the students earlier about the importance of environment. And that's what the HPL was and is in its own right. A unique environment where you get the opportunities to learn and to grow. Now I'm going to talk a lot about clinical, but I want to take a minute to highlight the applied physiology work that has come out of the HPL. And these are papers that I have cited numerous times. I highlight them here today because Bill is featured on all of them. And you'll note that this is Bill's unique contribution to science, first authored paper on leg muscle metabolism back in the 70s. But we have a key paper here on elevated plasma-free fatty acids and insulin, and also a caffeine paper. And you cannot go wrong with a caffeine paper. It will always be cited. It's always topical. It's always of interest. And we have a number, probably one of the key papers that Dave ever published was this paper related to the fiber types of track and field athletes, which is one of the reasons I came to Ball State, to learn more of that work. Not only did we learn a lot about applied science, we learned a little bit of nutrition, Bill, the importance of the donut, and importantly, the importance of caffeine. So let me segue now into the science. And this is going to be more clinical work, and it's exercise as it relates to clinical. Our working model of human disease relates to nutrition, and it relates to exercise or physical activity. And inactivity, poor nutrition, does contribute to the development of obesity. We have a huge obesity problem here in the US and worldwide today. In excess of 40% of the population have obesity. In excess of 68% of the population are either overweight or have obesity. And that leads to a lot of chronic conditions. Insulin resistance is a condition in which insulin, the hormone insulin, is not functioning correctly. It leads to a lot of pathological problems on the wrong genetic background, and that's represented here, leading to conditions like diabetes, cardiovascular disease. We've got women's health issues like GDM in pregnancy, polycystic ovary syndrome, fatty liver disease, and a whole host of cancers. I could continue to list numerous other conditions that related to this. All of this trails back to exercise, inadequate amounts of exercise, the way we've engineered activity out of our daily lives, and then nutrition, abundance of nutrition. And there's a lot that I'm not going to focus on nutrition today, but there's a lot we can say about the role of food in the contribution to this disease. This is a cartoon really to represent these two contributing factors, and I think it sort of reflects probably many of us in the evening time. This is an interesting slide. For those of you who can see this, if you can see it from the back, I think we've got 1950 serving sizes, and this is 20. I mean, they have changed a little bit in 2023, but not that much. And so we're looking at huge increases in serving size. And so we have more calories coming in, we have fewer calories expended. We're going to have an energy imbalance. And you're all bioenergetics folks, so you understand what that means in terms of weight gain and obesity. We've been fighting some of this, so I've noticed that some of the muffin sizes have reduced, but we still have this huge problem related to driving obesity. These are some of the medical complications associated with obesity. And I keep a big poster of this in a conference room when I meet with people, and they just can't take their eyes off it. The number of conditions that are associated with having obesity is immense. Now, I'm going to focus today on diabetes, but you can see a whole host of conditions. And I want to draw your attention to cancer as well. 13 cancers are now associated with obesity. And so we have even diseases that we would typically not associate with obesity having major contributing factors. I've got 3 segments to this. I'm going to talk about prevention, treatment, and cure. I'm going to start with diabetes. And this is a pyramid scheme meant to represent how we treat obesity. BMI is the term that's used to screen for obesity. It's your weight divided by your height squared. And typically anything above 30 is considered having obesity. Most of this group probably looks like well under 30. Hopefully some of you are under 25, which is what we're looking for in terms of normality. But as weight increases, BMI increases, and to treat that, lifestyle modification is usually pretty effective. People don't adhere very well, but it is effective. At another level, pharmacotherapy, drugs, and as BMI goes up and the conditions get worse, then we have to resort to treatments like bariatric surgery. And I'll share some of that data with you today. This is an important slide. It goes back to the early 2000s. This was the Diabetes Prevention Program study. This is a study that came out of Pennington Biomedical. It was the first study to demonstrate the effectiveness of lifestyle in reducing morbidity and mortality in type 2 diabetes. People were shocked by this. Metformin is a common drug that's used to treat type 2 diabetes. And you can see here's the placebo control, here's the metformin group in yellow, and here's the lifestyle group. Way outperformed the drug treatment. Over a 4-year period, continued over time. There's a continuation of that study today. It highlighted, really reinforced at the time, the importance of lifestyle, of exercise and diet in treating type 2 diabetes. But I'm going to focus more on the exercise mechanisms rather than the sort of epidemiological data or these large-scale clinical studies. The mechanistic data show us that there's improvements that can be achieved in terms of diabetes at multiple levels. We see it in muscle, we see it in adipose tissue, we see it in the vascular system, in the heart, and it leads, there's various pathways that are associated with this, and I know that all of the students are familiar with these pathways. Many of you may be charting them in your research. There's multiple efforts going on to develop drugs around these. We've done exercise studies to show improvements in how these pathways signal to each other, and that helps us to understand the improvements in insulin sensitivity and insulin secretion that's associated with diabetes. So let me define diabetes. Diabetes is a condition where you have high blood sugar in excess of 100 milligrams per deciliter at fasting. You take a glucose drink and your blood sugar levels go up to the order of 200 or 300. Typically, a type 2 diabetic will run blood sugars in excess of 200 to 300. Insulin is not working, so you've got insulin resistance. That prevailing high blood sugar causes multiple problems in terms of your organs, your retina, your nervous system, your kidneys, and it eventually contributes to hardening of the arteries and cardiovascular disease. And so if you can make insulin work better in the system, particularly in relation to getting sugar into your muscle, then you will improve that condition. And exercise, we find both resistance exercise, aerobic exercise, or any combination thereof, has significant improvement on insulin sensitivity. It also improves how your beta cells work. So your pancreas makes insulin, and in a diabetic, pancreas is not making sufficient insulin to keep your blood sugars under control. One of the things that exercise does is improves how the beta cells actually make insulin and secrete insulin into the circulation. And that's one of the studies I'm going to go into a deep dive, particularly for the students in the audience. You will also find that liver glucose production is affected by insulin resistance. So your liver is putting out excess sugar. That keeps your blood sugars up. The effect of exercise is to reduce hepatic or liver glucose output and to increase overall how glucose is working. This is an early study that we did when I was a postdoc in St. Louis using euglycemic hyperinsulinemic clamps, a very sensitive technique for measuring insulin sensitivity. And we have here insulin infused at 2 different doses, and the black bars represent the improvements we see in just 7 days of exercise in older, heavier individuals with obesity and with type 2 diabetes. So the effect of exercise is pretty quick in terms of its effects on, this is reflecting effects on muscle, and it also reduces hepatic glucose production, the liver output of glucose, both at rest and in response to insulin. So everything's working better here, very, very quickly. How about longer-term exercise? So here we have 3 groups: normal, those with prediabetes, and those with diabetes. Again, these are 60 to 70-year-olds. They have obesity, and they're falling into one of these 3 groups. Here's what happens with 3 months of exercise training. And essentially what you're able to do is you're reversing them. So these guys here who were impaired before exercise training are now normal compared to here, and these guys are back to impaired compared to here. So exercise alone, no dietary interventions here, no drugs, just exercise, has very profound effects on your insulin sensitivity and your ability to control your glucose. But the one question that sort of has been hanging out there forever is, if you think about exercise, really, what does your pancreas do? So it's in here, it's making insulin, it's not responding to exercise, it's not contracting. You know, exercise, you think of muscle, you think the heart, you think the lungs, even the liver. But the pancreas, what the heck? It's a very gooey organ, it's kind of not very formed. How does it respond to exercise? And the beta cells that are in there, how do they get better as a result of you exercising in the gym? So that's a question that sort of remained unanswered up until, well, we think we have part of the answer. But August, we presented or published this paper in Diabetes. Diabetes is one of the top diabetes journals. We were fortunate to get the cover of Diabetes for this study. And I'm going to share some of that data with you. And in that study, as a preparation, we developed a model. So when you look at physiology, we typically, in applied physiology, we're dealing with mostly humans. You can have preclinical models of a mouse or a rat, or you can go even deeper and get back to cells. Now you're dealing with bench foundational research, but that's how you discover the actual mechanism. It's very hard to discover a mechanism in a fully functioning human. We take muscle biopsies and we do our best with that. But you don't control and you can't manipulate all of the conditions as you can do in a cell. So science progresses from these basic cell-based studies all the way through out into implementation science in public health. And we are today focused here on that cellular model. But how do you do that in a muscle? Well, we took a muscle biopsy and we take out the cells. We can grow these cells into primary myotubes from the stem cells that are in the biopsy. And that's what that looks like. So these are our primary myotubes that we can then stimulate to contract. And you can see these cells, these myotubes, contracting at different frequencies. So you can change the stimulation. You can make them contract quickly or slowly. And this is a pretty unique model. We developed it over a long period of time. It took a lot of physiology. It brought me back to a study I did here at Ball State with Mike Flynn, where we took rats and we were doing these contraction experiments with equipment that we had to borrow from physiology. It was a laborious study, but it actually taught us a lot of physiology and a lot of information on how to contract a muscle in vitro. And using that knowledge, we applied it to this model, and we use this model then to simulate exercise. So this is basically exercise in a Petri dish. And we call this electrical pulse stimulation. So we've got muscle here in a Petri dish, which we've grown out into myotubes, and we stimulate it electrically, and muscle contracts. We take off the conditioned medium that it's sitting in, and then we measure glucose-stimulated insulin secretion in that medium. And that's what we show you down here. We see that this is insulin secretion from the cells that have been exposed to the media that has been contracting. So there's something in this media that's causing these beta cells. They're called, these are BTC6 cells and MIN6. These are just a type of beta cell. Doesn't really matter. They're a beta cell. They're seeing stimulation. They're in beta in a solution that has come from the muscle that's been contracting, and now suddenly they're releasing more insulin. So that told us there's something coming out of the muscle, being secreted, that is responsible for this effect. So then our job is to find that. Well, that's not that easy because there's a lot of stuff that comes out of that muscle. We did this through an RNA sequencing experiment where we identified all the various RNA sequences within the sample. There are thousands, literally. We screened them. There are various tools available for that. We're using these three databases here to filter it down. We bring it down to 71. This is work that was performed by Hannah Zhang and Stefan Newith when we were in Cleveland. So all of this work was done in Cleveland. We brought that to Pennington to finish. This is some of the work then getting down to identifying what is the best target. It could be any one of those 71. We went with GDF15, which on a scatter plot was identified out here. It was identified in this heat map. So we took a guess. This is your best guess. You can't do them all, and you have a lot of work ahead of you in terms of experiments that need to be done. I'm only showing you the tip of the iceberg here today. This was 5 years of work, and it led to the discovery that GDF15 is a myokine, what we call a myokine secreted from muscle, that has an effect to stimulate insulin secretion. And we show that here. Here's the GDF mRNA increased in two different ways. We see an increase in the protein in the conditioned medium, showing that GDF15 has increased in the medium that has been exposed to the contraction. And we show over here, I'm going to have to get my glasses out now. This is GDF in skeletal muscle. So we had some samples from other studies that we had done in human studies, like the one I just showed you with the humans impaired type 2 diabetics. So we took that muscle and we measured GDF15 in the muscle before and after exercise training. And this is the profile that we get from those individuals. So again, it supports the idea that exercise is doing something to GDF15 within the muscle. We've shown in vitro that that is being secreted out into the medium, and we've shown that in vitro that medium stimulates insulin secretion from the pancreas. So we had to convince the world, of course, that, yeah, that's a real thing. And here we have a series of experiments to show that GDF15 is not only there, but it is required to potentiate contraction-induced glucose insulin secretion. So not only does it happen to be there, if it's not there, you don't get the effect, or you don't get an increase in insulin secretion. And that is, this keeps jumping on me, sorry. That is what we see in these experiments. And I'll just highlight this one here. So this is no EPS, and this is insulin secretion. And then you can see with the EPS, you increase insulin secretion. And then we use the neutralizing antibody, so you can block out that GDF15, and you don't get that effect. It's like what you saw with the non-EPS, but this has EPS. So it's been stimulated. But when you block out GDF15, you don't get that increase in secretion. And then the non-EPS and the neutralizing antibody shows that this is comparable to the blue over here, and it shows you that, yes, indeed, this is a GDF effect. And these other graphs that I'm going to show you here all reinforce that idea that there is an increase that requires it requires GDF to get this increase in insulin secretion. We did that in mouse muscle, and we did it in mouse islets, and then we did it in various conditions in which we were able to work with a colleague out of Michigan, Randy Seeley, with his receptor knockout mice to demonstrate that it doesn't actually work through a traditional mechanism. This GFRAL is the receptor that's responsible for GDF. GFRAL doesn't exist in the beta cell, so it's working through an as yet to be identified mechanism. And we postulated in the paper some of the potential reasons for that. How about the physiology of this? Well, we used a number of measures here to demonstrate that these cells that were exposed to electrical pulse stimulation had greater glycolysis. The ATP to ADP ratio was increased. We used experiments to stimulate calcium release here in panel D, and you can see again with the GDF-15, all of that is increased. These are all the things that lead to insulin secretion within the beta cell. So these are all the steps that you need to have in order to facilitate insulin secretion. And so what we think is happening is that GDF-15 is increasing glycolysis in the beta cell, increasing this ATP to ADP ratio, increasing calcium, leading to insulin secretion. And that's schematically represented here, where exercise leads to GDF-15, an increase in beta cell function in the pancreas. Under glucose-stimulated conditions, then, you get an increase in glycolysis, increase in mitochondrial function, and an increase in ATP-ADP ratio, ultimately an increase in calcium, and eventually an increase in insulin granule release from the beta cells. So this was our story around GDF-15. It is a novel finding in and of itself. When we did the first experiment on GDF-15, there were, I think, 21 publications in total in the literature at that time. None of them were related to metabolism. By the time we finished this, I think there's about 3,500 publications on GDF-15 because the Novo guys recognized that GDF-15 actually has effects on body weight as well. And so there was a lot of development of the field that happened right after this. But it was a very unique experience for a lab to really discover something for the first time and then have to figure out how to get it to this point and really work in the dark. We didn't have all the tools. We had to develop those tools. It took us a long time to do it, but we persevered and we made that happen. And we have made this seminal contribution to the field in terms of understanding how exercise contributes to improvements in beta cell function. So let me move away from that story now to obesity. And I don't know how many of you here know about Wegovy, Ozempic, or Mounjaro, but this is, in Irish vernacular, this is the skinny jab. And the skinny jab is a drug that reduces body weight. It has been developed by Novo Nordisk out of Denmark. And Julene may be interested in this story, of course. She leads the Novo lab in Copenhagen. But we have Ozempic and Wegovy. Ozempic was developed for type 2 diabetes. It's a GLP-GIP. These are incretins. I'm going to tell you what they do in a minute. They had profound effects on curing or reducing diabetes in patients with diabetes, but the effect appeared to occur because of weight loss. And as a consequence of that, people started taking the drug not because they had diabetes, but because they want to lose weight. And this is a story that's across the media today. Novo drive, I think, up to a half of GDP in Denmark today based on sales of Wegovy and Ozempic. Here in the US, this is selling for $1,500 a month treatment. There's big issues in terms of access. It's a very effective drug. Here are two data sets that just came out. This is not Wegovy. This is two new drugs that are not approved by FDA yet. tirzepatide. Both actually are Lilly drugs. I met with the Lilly folks yesterday. They're very excited about this particular drug, but you can see here, and I just want to show you two pieces of data. This bar here represents a 20% weight loss with a year on that drug. So you think about that, 200 pounds, 20% weight loss, 40 pounds. That's a heck of a weight loss to be able to maintain that over a long period of time. The most effective drugs that we have on the market today. They're called incretin drugs. They are made up of a combination of two peptides, glucagon-like peptide and GIP. These are gut peptides that are secreted from the gut primarily to regulate your metabolism. The effects on satiety were unknown before all of this discovery, and they're working primarily to reduce satiety. So they go signal from the gut to the brain. You don't eat as much. These people have very little appetite, so they eat less and lose weight. Other effects we'll see in a minute, but that's primarily the mechanism of action. And you can see here there's another drug that is producing 25% weight loss, which is now comparable to what you get with bariatric surgery. And so these are this is a whole series of drugs that are really transforming the obesity field. These drugs, GLP-1 and GIP, work through these mechanisms. I just want to highlight satiety. I want to highlight gastric emptying is slowed, and it has effects to increase insulin secretion. The second drug that was on that panel has glucagon in it as well, and that enhances glucagon secretion, helps to regulate your blood sugar, and that in turn helps to lose weight. So these are really profound drugs. If you have not been paying attention to this will change the landscape in how medicine is practiced. Because go back to the first slide on the model. Obesity was at the root of all of these other problems. And then you look at obesity and all the multiple diseases associated with that. You treat obesity, suddenly you're treating cardiovascular disease, diabetes, fatty liver disease, hypertension. And so I don't know what's in store for the pharma world, but if you treat with one drug, then you're not treating with the other 6 drugs that are used for these various conditions. So it's going to have a very profound effect. For those of you who have had experience with it or have taken it yourself, you'll know the impact it has on your weight. Well, like all drugs, there's been many drugs that have come down the pike for obesity. Many of them, they've all disappointed so far. There's always some side effect. The side effects with these drugs to date are minimal. It relates to primarily gastric issues, but there hasn't been a big showstopper in terms of, we can't use this drug yet. Will it happen? History says yes, it will happen, and that's what I'm banking on. But it might not. This could be really revolutionary. As the bioenergetics guys, as all of you are, I'll remind you, this is ATP production from glycolysis and oxidative phosphorylation. This is a process that is very tightly coupled in mitochondria, and I use the word couple as an introduction to another potential medication or form of drug treatment for obesity. That is the uncouplers. So we have been using this mitochondrial uncoupler for the last few years in an attempt to understand, are there ways for us to control blood sugar? Are there ways for us to control obesity body weight independent of or using a drug approach? And this is a chemical compound called BAM15. It's a lipophilic weak acid. It is not easily incorporated into the system, but you can put it into food. And so we've been doing a series of studies. I'm not going to go into the data in detail because these are published, but we have been able to use this uncoupler in preclinical models now, in mice where we feed mice who have been fattened up on a high-fat diet. And what we find is that we can change their body composition. We can improve their blood glucose. They don't gain weight on this drug. So they're eating a high-fat diet, but they're not gaining weight. It's driving energy production by uncoupling energy, burning up essentially calories within the system. It typically goes to fat. And so it's in the fat and it's burning energy there. And none of that, it doesn't raise core temperature, which is an important aspect. And it has a profound effect on muscle, increases fatty acid oxidation, it increases glucose uptake, and all the good things that you expect muscle to do. And all the bad things that you associate with adipose tissue is reduced. A decrease in fat mass, a reduction in de novo lipogenesis, and an increase in AMPK phosphorylation, a key enzyme in the metabolic pathway. And then in the blood, we see a reduction in your fatty acid levels and your glucose and your insulin. So it's very effective. And that we've tried this now in an obesity model, worked very well, published the data. You can read that. We did a cancer study, and so working on the mitochondria and the cancer cells, saw a reduction in the incidence of the cancers, in specific in breast cancer. And then we did this study in aging. And so you might appreciate this a little bit more. This is a muscle study. And so one of the things we were seeing was that muscle mass was preserved in these mice. And one of the, I didn't get into defects associated with the GLP-1s, the Wegovy's of the world, but one of the things is as you lose weight, you lose muscle. And so with advancing age, you really don't want to lose muscle. So that's a counter for using these incretin drugs. But this drug, we find that you actually preserve muscle mass. So we set about an experiment in aged mice. So we had mice that were 80 weeks old, which is pretty old, and we put them on a high-fat diet, and we measured their body composition, we measured their muscle function, and we measured their muscle composition. And sarcopenic obesity is a condition that sarcopenia, some of you will be very familiar with, loss of muscle mass with aging. Sarcopenic obesity is the association of loss of muscle with weight, with an increase in adiposity. So you gain more fat, but you don't gain muscle. You develop this condition. It starts at about the age 40 and progresses as you get older. And what happens is that you get inflammation, you get a reduction in insulin sensitivity, it acts on the mitochondria, and you have a lot of negative effects that are leading to degradation of the muscle. And you have a loss of muscle function, and you have a loss of muscle mass. When we feed these mice this BAM15, we target the mitochondria. We find that we can reduce various enzymes associated with cytochromes. Apoptosis goes down, and we see a reduction in inflammation up here, leading to a reduction in protein degradation, the loss of protein. And these mice then have actually not just more muscle, but when you put them through a physiological test, and the way we test mice is we put them on a bar and have them hang. And the stronger they are, the longer they can hang. And these mice who get the BAM15, they can hang for a long time. Not only do they have more muscle, but it's working better. And so we're really very excited about that discovery. Long way to go with BAM15, long way to go with these uncouplers. They have a history, some of you may know that, but this is an area that we're pursuing as a new treatment for obesity. Let me segue now to the last piece of the story around obesity and treatment, and that is to bariatric surgery. Now, bariatric surgery over the last, I would say, 20 years has really come into vogue because of the effects it has on metabolic diseases. It's no longer called bariatric surgery, it's called metabolic surgery. And that's based on the observations that bariatric surgery reverses type 2 diabetes. So you can take a patient pretty much like this individual here. A lot of these folks who are going for surgery have BMIs in excess of 50. We did a lot of this work in Cleveland, and the largest patient had a BMI of 85. So these are really, you come from a lab like the HPL and you think, oh, exercise. If you only exercise, you'd be fine. You go into a floor where you have patients with BMIs of 50, 60, 70, and you very quickly realize exercise is not going to fix this. And so we have these medications, but at that time we didn't have the medications, so surgery was the first line of defense here. And it's often multiple surgeries. So I worked extensively with the bariatric team. We had various surgeries. I worked with one of the top, Phil Schauer, one of the top bariatric surgeons in the world. And we predominantly were doing these Roux-en-Y gastric bypass, the gastric bypass procedure. That led to the discovery that bariatric surgery reverses type 2 diabetes in obese patients. We published that in the New England Journal of Medicine in 2012, kept following those patients. This was the first randomized trial to show this outcome. Followed these patients for 3 years. Another New England Journal of Medicine publication. New England really liked this stuff. We went back in 5 years, same patients. We published again the observations that bariatric surgery is effective, it's safe, and it's durable in terms of the reversal of type 2 diabetes, even in patients who don't have a BMI of 70 or 80. So we had patients in here, the lowest BMI was 28. So there's people in this room who have a BMI of 28. This patient had severe type 2 diabetes, and it was intractable. Went through the surgery, reversed the diabetes. That's not a weight loss issue. This is a guy who didn't lose a lot of weight. This is something that's happening with the surgery. And so there's a lot of work going on right now in terms of understanding that. One of the outcomes of that, there were a number of centers across the country who were doing these randomized clinical trials, the Joslin in Boston, Pittsburgh, University of Washington, 4 sites. We came together, formed a consortium, developed this ARMS type 2 diabetes trial, got funding for this, and I've been working on this since I left Cleveland. We just submitted the 7 to 12-year data, and it looks like it's going to get accepted in JAMA. So we're pretty excited about the findings and the quality of the work. But this is the 3-year data from that study. And what it shows is that the way you measure improvements in diabetes is with your HbA1c. That's your hemoglobin A1c. And typically it should be around 6.5%. It's glucose that binds to protein in your blood. And the higher your glucose, the higher that number is going to be. And the goal is to get it down to 6.5%. Now, what's unique about this trial is, so when we did the studies in Cleveland, it was a small cohort. And so we're criticized, well, yeah, you've got these, we had, I think, around 120 patients. But you need more patients. And you need to show that it's not just in Cleveland, that it's in all of these sites. So we brought all of these folks together. We had something like 380 patients to start with. It's the biggest trial of its kind to date. And at 3 years, we were able to show significant improvements in blood glucose control, HbA1c, significant reductions in body weight. And you can see here they're down around 20-30% body weight. So that's fairly close to what I showed you earlier with the drugs. So the drugs are actually now approaching this kind of level. And I should say, typically with lifestyle intervention, if we get to 5% weight loss, then we consider that a success. So that's the difference here between what we're seeing with these drugs and with bariatric surgery. But the key for these studies is it durable? So typically with weight loss, you rebound, and once you stop exercising or you stop following the diet, then you're back where you started. Well, you can see with bariatric surgery, you're pretty much locked in on this. Now, this study went out for up to 15 years, and we have patients who are 15 years out who had type 2 diabetes at the beginning, who no longer and have not had type 2 diabetes since. It's an interesting thing in medicine. You can't say they're cured. It's a chronic disease. But if you've not had type 2 diabetes for 15 plus years, then you feel cured. You may not be categorized as cured by the profession, but you have certainly sustained remission. And so that's the term that we use. A lot of the effects, these patients are taking a lot of drugs. So this is what it looks like before the surgery. They're on a whole a variety of insulin and various drugs. And you can see that for up to 50% of the patients who undergo bariatric surgery, they're off drugs. Now, the important thing about this is not only are they off drugs, they leave the hospital without the drugs. So they come in on medications that are controlling their blood sugar. They're running maybe 200 milligrams per deciliter. They have the surgery. They're usually released 48, 72 hours after surgery. They go home without meds. So something has happened, and when you replumb the system, that has changed how the body is responding. It happens very quickly. We really don't know how or why. Now, over the last 20 years, we were working on GIP and GLP-1 as the potential targets for that. And a lot of the research that was driven by bariatric surgery contributed to the discovery that these drugs actually cause weight loss and regulate the gut-brain axis. So we contributed indirectly to what Novo and Lilly have today. But you can see here, for the surgery group in particular, a huge reduction in the numbers that are not taking medications. Not so for the medical surgery group. So this is a randomized study. So some patients didn't get surgery at all, and we still follow them. And you come into a study, this is difficult, as you can appreciate. You've got patients who, you would prescribe the surgery, but you want them to participate in a long-term trial, and you're asking them to not have the surgery. And so we have a group of patients that we have followed now for up to 15 years who still haven't had the surgery. We have had some crossover, and the ones who cross over obviously do better, but we've had a lot of patients who, I don't know, it's one of those things where, they get into the study, they know that this is working, but for whatever reason, they don't opt to take the surgery. Is it safe? It's as safe as an appendectomy in terms of complications. And in terms of mortality, it's as safe as knee arthroplasty. So we would say that it's relatively safe. That's an issue that a lot of the public perception has to address. And these are the mechanisms that we've published on this as well. That's something that you can read. But we think that it's these gut peptides or something. It doesn't necessarily have to be GLP-1 because we've actually eliminated GLP-1 and GIP as our candidates for the improvements in insulin secretion from the pancreas that we see in these diabetics. We think that there are some other unidentified proteins that are responsible for this. And we're in the midst of that experiment I showed you at the beginning where you do all of that RNA-seq and you follow it. It takes a long time. That's what we're in the middle of, trying to identify the potential target. So that's the end of the basic science session. And I want to segue into just a couple of minutes on more of a public health message. I was involved with a joint international consensus statement a few years ago on the stigma of obesity. You really have to understand that there is a lot of bias. In this room, there's bias in terms of obesity. There's a lot of stigma associated with it. And so in order to be able to address all of that, we have to recognize that we have this issue. So we have this international group that wrote a really nice paper if you want to understand a little bit more of the stigma of obesity. But just to put this into context for you. So the problem is a lot of perception. So if I said to you, you have cancer, now suddenly you're a victim. But if I said to you, you're obese, well, that's your fault. So you're a culprit. That's how we think about this problem. And so we've thought about that way for a long time. So for people who are living with obesity, they've been judged, they've been blamed, they've been shamed, and we need to change all of that. And so this change is only going to happen when we recognize that obesity is in fact a disease. So you got to tell a different story. So we got to move away from this perception that it's your fault to it's a disease. Eli Lilly and Novo Nordisk would not be making a medication for something that wasn't a disease. They have made the medication. It is working. This is a disease. So we created this was a national public service awareness campaign. We went out and raised, I think, this cost about $1 million. We raised $1 million through philanthropy. Created this campaign called Welcome to Obesity, deliberately misspelled, USA, the state of America. And we created various campaign elements, including a website. We had these out-of-home media that mimicked roadside destination signs and radio messages. These were all inspired by state tourism approach. So these are actual billboards that appeared across the country and in major metropolitan areas in New York, Chicago, and LA. And some of the messages were a little tongue-in-cheek. Obesity, where fast food restaurants outnumber grocery stores 6 to 1. Sounds odd, but that is actually a fact. Where 1/3 of our TV ads are for junk food, another fact. Where you can have dessert for breakfast, we call it a muffin, absolutely a fact. And so we also created, I'm going to share with you a couple of videos here. These take 1 minute each. These videos were designed to highlight the complexity of obesity and to tell the story of kids with obesity. The sort of stigmatization, the bullying, everything that goes on with that.
3,
Charlie, you're gonna be
4.
late.
Charlie.
Hey, Invisitor! Wait!
Things are slow at work.
We can't live like this.
We were able to do this as a nonprofit, and we had a company who promoted all of this. TV directors selected these for showing. We had them show before Saturday Night Live, among other shows. Here's the second one.
Well, when it comes to your weight, I'm going to tell you. Yes, who's next on a scale that never fails?
What about me? Can you guess my weight?
Well, when it comes to your weight, I'm going to tell you what I know. I know the healthier option often isn't an option. That you don't exercise enough because you can't. And how everyone blames you, especially yourself. I know your weight is out of control because I know it's not just what you're
life
eating,
of these kids.
it's what you're being.
And the idea here was to work together to solve obesity. These were quite successful nationally. It was in excess of 1.2 billion impressions all across the country. We raised over $14 million in media for all of this. But most importantly, what next? So what next for us was grow healthy. Deliberately playing on the Louisiana spelling of grow, G-R-E-A-U-X, and focusing on childhood obesity. We convinced the state of Louisiana and the governor to provide $25 million to support this Grow Healthy initiative, which is actually building on all of the programs that have been developed at Pennington and across the country over the last 30 years. Focus being on childhood obesity and reversing, educating, and changing the dynamic. I want to finish with, I'm going to lose my voice here. I'm going to finish with a thank you to Bill. You know, when I came here, I was asked, you know, what was the most memorable event in your time? And this is probably the most memorable event because we had a preemie in Ball Memorial Hospital. It was one of the most, it was a traumatic experience. But I want to say thank you to Bill because Bill was Brent's godfather. And I want you to know, Bill, the picture on your right is Brent with the next generation taken just a couple of weeks ago. And so this is a kid who's grown into an adult and has been very successful. I want to thank Bill for being his godfather. Excuse me. So with that, as my voice fails, I'm going to hand off to Mike Flynn. Happy birthday, Bill, from me, from everybody here. Thank you.
Thank you, John. That's the first lecture in the series. You'll be a tough act to follow. Good afternoon, everyone. My name is Mike Flynn. I'm a proud graduate, 1987, from the Human Performance Lab. It's my honor and pleasure to be here. My job today is to close out the talk with a short recognition of Bill Fink on the occasion of Bill's 90th birthday. Happy birthday, Bill. Bill, I don't know if you remember, but I brought that plastic putting green into the lab. And that caused you to get the bug for golf again, and which led to you getting a membership at the Outs Club. I'm not sure Dave was really happy about that, but at least he knew where to find you. On a serious note, the impact you had on my career is immeasurable. You taking the time to teach me practical biochemistry techniques gave me the confidence to try anything. Probably some things I had no business trying. Taking the time to review and edit my written work forced me to become a better writer. And I know it'll make you laugh to know that I now make my living as a medical writer. We wanted to let you know how much your mentorship is appreciated. I had the honor of identifying a small number of esteemed HPL graduates who wanted to say thank you. We'll let them represent the multitude of individuals with similar sentiment. Enjoy.
Happy 90th birthday, and to let you know how important you and your teachings have been for my career and personal development. More importantly, your guidance and encouragement have shaped my efforts with my students over the last 40 years here at Iowa State University. In my 4 years as a graduate student at the HPL, you invested both faith and trust in this undereducated yet motivated student. Whether it was developing methods to measure muscle buffering capacity or to measure saliva viscosity, or even just washing disposable test tubes so we could reuse them, you gave me the confidence and opportunity to try new things in the lab. some of which actually worked. For that, I remain truly grateful for your mentoring. Thank you for all your mentorship, and I hope you have a happy 90th birthday. Thank you.
Greetings and birthday wishes from down under, Bill. I hope you have a wonderful day and enjoy the celebration in Muncie. I'm sorry I can't be there. It's 40 years since I was in the lab, but some of my fondest memories remain discussions on life, religion and science with you over the Turner fluorometer as I mastered my constriction pipetting techniques, lactate assays, glycogen assays, and the ill-fated QO2 assay, which caused some anxious moments for a while until it was validated. So thanks for some wonderful memories. Again, I hope you have a wonderful celebration. I wish you many more years of health and happiness, and hopefully we can catch up in person before too long. Bye-bye.
Bill, hi. This is Julene Zerath, class of '86. Remember me? Good. I just visited the Ball State website this morning to check up on you. When I got to your profile page, I noticed you now hold the esteemed title of Assistant to the Director Emeritus of Human Performance Laboratory. Geez, Bill, what an upgrade. Congrats. But do they still let you use the showers? Another thing I noticed was in the place where your photo should have been, there was a photo of Beneficence, the Ball State's institutional icon, which, as many of you may recall, is affectionately called Benny. I thought that was kind of symbolic, Bill, because beneficence is defined as an act of charity, mercy, and kindness with a strong connotation of doing good to others, including moral obligation. Kind of sounds like you, Bill. Maybe it wasn't a mistake that Benny was pictured. As Bill, we all greatly admire you for your beneficence. Happy birthday, my friend. Wishing you all the best for many more fast and steady miles. Take care.
Hey Bill, Brett here. Just wanted to say a belated happy birthday and to thank you for everything that you've done for me, as well as the HPL program over the years. You've been a super important part of the history and tradition, and frankly the success of all of us as we have gone out in the world and tried to make an influence based on what you have instilled in us. And again, just want to say thanks, and I hope to see you soon.
Bill, greetings from the Pacific Northwest. This is Leo here. I'm getting my exercise in, and based on what I'm seeing, I'm just north of 200 watts. Wait a minute. I think I'm a little off. Glasses are dirty. Oh, just north of 100 watts. Sorry about that, Bill. Bill, you've been a great inspiration, not only to me, but to many students that have gone through the HPL. I can't thank you enough. I was a student there, of course, and have visited over the years. We have another student here that wants to say happy birthday. Here we go.
Hey Bill, Jared here, wishing you a happy birthday. Hope everything's going well. And time for me to hop on the bike and join Leo.
Here we go. Happy birthday, Bill. Buon compleanno. 100 anni di salute. Wishing you strength and health. And thank you for everything, Bill. Ciao, ciao.
Bill, and wishing him a happy birthday.
Well, thank you, John, for a wonderful lecture, and Mike for leading the portion on Bill. Bill, again, happy birthday. Thanks for everybody for coming. If you have some follow-up questions for John, feel free. We're going to just roll into a reception now, so feel free to help yourself and mingle. And thanks again for coming. We greatly appreciate it.
Yeah, sure. No, I have no permission. I do graphic